Hi everyone. Welcome to our first FocalPlane Features Meets preLights webinar. So this is a brand new collaboration that we're trying out. So first to introduce myself, I'm Helen. I'm one of the community managers here at the Company of Biologists. We also have Anatoly on the call, who I think you can't see because he's not talking at the moment, but you'll see him at the end. So he's also a community manager here at the Company of Biologists. That's who we are. Just giving you a quick introduction into what FocalPlane is. So FocalPlane is a microscopy community site. I'd really like to encourage you all to have a look and to participate. So one of the important things to tell you is that anyone from the microscopy community can contribute to the site. And that means that we have some wonderful resources on there to help out cell biologists or microscope end users in particular with their imaging issues all the way from sample prep or through to image analysis. We also have the classics like jobs board, events calendars. So yeah, check them out, and interviews with people in the field. So yeah, get involved. Let us know if there's anything that you'd like us to do. Have a look at our gallery, which is filled with some beautiful microscopy pictures. And yeah, generally reach out to us if you're interested in getting involved, and have a look at the website. Just a quick other plug that FocalPlane with the Journal of Cell Science has some microscopy training grants that you can apply for. So these are open to early career researchers in cell biology, and you can apply for a grant to attend any microscopy or bioimage analysis training course if you don't have funding already to do so. There's actually an application deadline tomorrow. So if anyone wants to attend something very soon, the application process is pretty simple. So yeah, have a quick look at that. So that's everything from me. So I'm just going to pass over to Felipe and Vibha, who are two of the pre-lighters from our preLights community site. And so they're going to tell us a little bit about preLights. Also, they're going to give their live preLights, so their highlights of a preprint that they've read, and then they're going to be introducing the researcher who did the work on the preprint. So yeah, we're excited to hear from them all. So yeah, I guess over to you, Felipe, I guess first.
Thank you, Helen, for your introduction to the FocalPlane community. I'm just going to share a quick slide here about before diving into today's paper. I just want to highlight the briefly the FocalPlane sister community, preLights, that is hosted also by the Company of Biologists. This is an interactive hub, as you can see here, between the ECR community. preLights is this community run by early career researchers who select, highlight, and comment on exciting papers across biology and provide critical perspectives, spark discussions such as this one, and help researchers navigate the rapid growing preprint landscape. preLights also helps to share preprints in life sciences, events, job opportunities, and expert perspectives, making it a go-to place actually for staying engaged with the latest in cell biology and its related disciplines. As a preLighter myself, I encourage everyone at an early or late stage of their careers to join this amazing space. I've had an amazing opportunity to discuss incredible preprints that are now actual papers, published papers with renowned authors, and ask them questions that I'm sure cross everyone's minds when you're reading a preprint, like eager to know more about this beautiful writing they do. Later, my co-host Vibha will tell you a little bit more about this initiative. And without further ado, we are here to discuss with first author Carmen Rodilla-Ramírez from Dr. Bottanelli's group at Free University in Berlin. Their fascinating preprint T-cell protrusions enable fast localized initiation of CAR signaling. This preprint explores the intricate mechanisms of T-cell activation at the earliest stages of cell-cell contact. I particularly chose this work because it provides a crucial quantitative framework beyond the static observations that we're used to see to capture dynamic rearrangements of the signaling proteins and the structures that help develop the T-cell immune synapse during CAR-mediated activation. I was also particularly drawn to this because I did my PhD in B-cell immunology and immune synapse, so it's incredibly fun to see this amazing work. And since we're doing this with a focal plane inspiration today, I would like to particularly highlight the imaging methods and innovation this paper provides, especially in microscopy and the pipelines for analysis. So please welcome everyone, Carmen, and let's hear about her fantastic work.
Hi, I'm going to share screen. That was a very nice introduction. Thank you so much. Okay, so can you see the screen? And can you hear me? Okay. So thank you so much for the introduction. I'm Carmen and I'm currently finishing my PhD at the Bottanelli lab in Berlin. And I'm very happy and very thankful that I can discuss the preprint that we have just submitted that is entitled T-cell protrusion enable fast localized initiation of CAR signaling, as Felipe said before. So I guess that the background of the audience was going to be quite broad. So I'm also going to do a broad introduction. Basically, I'm studying T cells, which are very important immune cells from the adaptive immune systems that help protect us from threats like virus infection or cancer. And they do so through their T cell receptor, which is here. The T cell receptor is a multi-subunit complex that specifically recognizes sites that are presented by this molecule that it's called the major histocompatibility complex. And it's in the surface of antigen-presenting cells or target cells. And when this recognition happens, the TCR gets phosphorylated by the first kinase of the signaling cascade, which is called LCK. And this process is actually reversible by the large phosphatase CD45. But anyways, when the TCR is phosphorylated, this allows the second kinase called sub-70 that is normal in the cytosol to translocate into the active sites of the TCR, which are called items. And it will eventually lead to the activation of sub-70, which will be able then to phosphorylate the first adapter of the cascade, which is called LAT, that will recruit a bunch of different factors. And from there on, the signaling cascade branches out, and it eventually needs to upregulation of some genes and an overall phenotype change of the T cell. So cancers have developed, obviously, a way of being able to avoid the immune system, and a way in which they do so is by messing up with the presentation of the peptide by the MHC. So scientists came up with an amazing idea, which is to use chimeric antigen receptors, or CARs. And these are chimeric receptors that combine the active signaling domains of the TCR with a fragment derived from an antibody. And now the T cell is able to, instead of mount a response against a PMHC, to mount a response against a specific protein. For example, in this case, we are using a HER2 CAR, which recognizes the protein HER2 that is upregulated in some types of breast cancer. And we are using a second generation CAR also, which also additionally has some costimulatory domains. Okay, so I don't know if you noticed, but I'm representing T cells like a very round ball here. And this is actually how normally people draw the T cells in textbooks, like for example, the Janeway. But actually, T cells are covered with a very heterogeneous and rich set of actin-rich protrusions, and they have been widely studied in the context of, for example, T cell migration. However, recent studies of the last decade or so have pointed out that they also have an important role in T cell activation. So for example, they are able to scan the opposing surface of a target cell very quickly because they are very dynamic, and they scan it in 1 minute. And they have also been shown to push into the target cell and make the membranes get close together. And when you disrupt the actin cytoskeleton with, for example, some drugs, you can observe how the close contact formation with a lipid bilayer is delayed. So the aim of this study was to understand how this was translated into protein rearrangement, and specifically how it's translated into protein rearrangement in a cell-cell contact and that it's CAR mediated. So normally, localization of the key signaling proteins that I have introduced is done through immunostaining or expression of tagged constructs, but we wanted to monitor this process live which and also avoid the artifacts that are normally associated with these techniques. So for this, we use CRISPR-Cas9 gene editing and to endogenously tag our proteins. And the protocol that we followed is actually just published in this preprint in this paper that my colleague Petia did here. So in case you want to check it out, I do some promotion. And basically, we use this to endogenously tag with a halo tag. I'm going to repeat the proteins: the phosphatase CD45, the kinases LCK and Sub70, and the adaptor LAT. And of course, we will express the CAR and also tag it. And we could successfully tag it, and you can observe how they are localized to the membrane, LCK, LAT, and CD45, while Sub70 is in the cytosol in resting conditions. This tagging allows us to observe the protein arrangement live, and also like with confocal and also with super resolution STED microscopy. And this is nice because we can see very fine details in these protrusions that are actually quite small in size and diameter. And then we can observe how they change when they contact the target cell, which is very nice. But in this next slide, I'm going to show how they are arranged before contact, because actually the localization of the key signaling proteins in resting T cells has been reported in controversial ways. So we wanted to systematically study it using our techniques. And we compare the distribution of these signaling proteins with an evenly distributed membrane marker that is basically a CAX motif that is tagged with a snap tag. And just by eye, you can see that the distribution of these proteins is not very different from the distribution of the membrane marker. But we used, of course, we want to put numbers into this and be systematic. So we teamed up with Amin from the Evers lab, which is in this building. And we created this pipeline for segmenting these protrusions. I'm not going to go much into detail into this, but basically we get a mask of the protrusions in the end of the pipeline. And with this, we can compute, for example, the enrichment of a protein, which is basically the normalization of the signal in the protrusions divided by the signal in the whole plasma membrane, and also the relative enrichment, which would be the enrichment of a protein divided by the enrichment of the membrane marker. So when we calculate this for our signaling proteins, the relative enrichment, if it's above 1, this would mean that the protein is accumulated in protrusions. If it's below 1, it would mean that it's excluded from protrusions. And if it's at 1, it means that there is no preferential localization to protrusions. So when we calculated it in our proteins, we saw that basically before activation, all the proteins were not preferentially localized to any part of the membrane of the T cell, like protrusions versus the main body membrane. But this changes upon activation. And this is what I'm trying to show here, but I'm going to walk you through this because maybe there are a lot of things happening in this video. Basically, you can see a JORCAT T cell that is expressing the membrane marker and the HER2 CAR, and a breast cancer cell here in magenta. So what we can see first is that the T cell is making contact in the beginning with the breast cancer cell through its protrusions. And the second thing that we can see is that upon this contact, there is a clear clustering of the CAR in the tips of the protrusions. This is what I'm trying to highlight here. In here, I'm basically just highlighting the protrusions in gray, and here it's the HER2 CAR, and here is the membrane marker. So you can see that before activation, both of them are localized more or less in the same way, but after activation, while the membrane marker obviously doesn't really rearrange, that would be bad, the CAR strongly clusters on the tip. So we wanted to follow this numerically on time, and we did this by segmenting the membrane of the JORCAT in 4 sets. So the first set would be the protrusion contact, so the contacts that are done through protrusions with the breast cancer cell. The second set is the contacts that are done through the main plasma membrane. And then it's the protrusions that are not contacting and the main body of the plasma membrane that it's not contacting. And we could see that we wanted to see what is the contribution of protrusion versus main body membrane to the contact. And we could see that the first 2 minutes of interaction were very protrusive. So in the beginning of the interaction, the immunological synapse is very protrusive. And in those spots of contact, there was enrichment of the CAR, both for the main plasma membrane contact and for the protrusion contact. However, the increase of signal of the CAR was faster and also stronger in the protrusions contact. So we wanted to also understand whether these early form clusters were activated units. So if they were already signaling, and if this enhanced CAR clustering in the protrusions was translated also to an increased recruitment of like the downstream the downstream molecules. So in here you can see sub-70, which is the kinase that was localized in the cytosol normally, but then translocates and is indirectly membrane bound when the CAR is phosphorylated. And we could see that indeed upon contact with the target cell and CAR clustering, we could see an increase of recruitment of sub-70. And this recruitment of sub-70 was enhanced in protrusions versus the main body membrane. So sub-70 is responsible for phosphorylating the first adapter of the signaling cascade, which is LAT. And LAT strongly clusters upon activation, which is known. And we could obviously see that LAT was clustered upon activation, meaning that activation was successful in our system. And we could see that in the same way as for sub-70, LAT was clustered more strongly and also faster in protrusions versus the main body. And interestingly, a thing that we could see is that the LAT clusters that were formed, they were formed originally in the site of contact, but they sometimes also segregated from it. You can see it now here. Now here. And we could confirm this using a super resolution STED microscopy. And this is what I'm showing here. Here you can see a Turcat cell that is making contact with a target cell that I'm not showing. And we wanted to compare the distribution of these clusters with the distribution of the membrane marker, just to make sure that it's not a random accumulation of membrane there. So you can see how the protrusion is nicely drawn by the membrane marker. And on the tip, you get a very strong cluster of the CAR receptor. And in that same spot, you get LAT clustering. However, you can also see LAT clustering throughout the protrusion, not co-localizing with the CAR. So it's clear then that protrusions are doing this better. So protrusions are accumulating CAR better, and they are accumulating the downstream activation proteins better. So we wanted to understand why, basically. And we then turned to see the positive and the negative regulators of the cascade, which are the kinase that phosphorylates the CAR, which is LCK, and the phosphatase that dephosphorylates it, which is CD45. And in here, you can see LCK. And basically, it was quite uninteresting because we could not see any strong rearrangement at the macro scale of LCK at any point of the interaction. And we could confirm this using STED. So in here, you can see images that were done in the same way as the one that I showed before with STED. And meanwhile, after contact, the CAR was strongly clustered in these protrusions. LCK looked more or less the same as the membrane marker. But this is not what happens to CD45. And CD45 is a very interesting protein because it has a large, actually, this is not even drawn to scale. It has a large extracellular domain that it's also very rigid. And it's like way bigger than what the complex of the CAR and the ligand is long. Okay. And so like from the site in which the CAR is contacting the ligand, necessarily CD45 has to be excluded, and this has been actually proposed to be the cause of CAR triggering or receptor triggering in general, also for the TCR. So we were interested to see whether we could see a difference of exclusion in protrusions and in the main plasma membrane. And what we could see was that from the very first moment of contact with the target cell, we could observe a decrease of CD45 signal. And obviously, if this was, well, obviously not. This was observable already at the level of confocal, but it was way more clear with STED, and this is what I'm showing here. This is the nicely drawn protrusion by the membrane marker and the CAR cluster on the tip. And in that same spot, there is a dramatic decrease of CD45. So we were able to monitor this over time. And here I'm showing the video because it's always nice to see the video I love to see these cells interacting. And we could see that the exclusion of CD45 is stronger at protrusions than in the main plasma membrane contact, and it happens basically right away of the interaction of the T cell with the target cell. So it seems like this increased clustering and increased recruitment of downstream effectors to the CAR clusters is happening because because protrusions are able to exclude CD45 more efficiently than the rest of the plasma membrane. And this brings me to the last slide. Basically, the conclusion of it was that actin-rich protrusions do enable fast localized initiation of CAR signaling because although before activation, all the proteins are localized without any preferential to the actin-rich protrusions, after activation, we get a more strong rearrangement of the key signaling proteins in protrusions in comparison to the main plasma membrane contact. So with this, I wanted to thank you all for listening. And well, I'm not going to summarize the acknowledgements. You can read that in the paper, but I will take any questions.
This is such a great work, Carmen. Thank you so much for your presentation. I'm just gonna allow myself to ask you a few questions.
Yeah.
Of course, videos are amazing. Time-lapse microscopy is great, but there something you wish you could have done with other techniques that maybe weren't available at your space or you could have done somewhere else? Is there any other thing that you would have liked to try, like longer times or exposures, if that's possible with your techniques and your approach?
I mean, I would definitely have wanted to do some kind of 3D imaging because I think this process, again, at the end of the day, we are focusing in one plane. So we are missing information that happens in the rest of the cell that it's not being focused in that moment. However, like with volume imaging, you also get a lot of photo bleaching and endogenous detected cell proteins, of course, are very dim because you don't have much. So this is tricky in general, but I would love to see them 3D interacting. And I mean, I think it would give also a strong point that this is not only a thing that happens specifically in these planes or basically I'm just going to say 3D imaging, super resolution 3D imaging if it's possible, because sometimes with other techniques you don't get such a nice resolution.
Yeah, it makes sense, even though your images are amazing. And in that same area, how do you feel developing this quantitative framework by using Cell Profiler? Did you try another environment like Fiji, Python scripts, or was it comfortable enough to do it in Cell Profiler?
Yeah, so I tried a lot of things actually now that I think about it, because when I started my PhD, basically I started right away trying to figure out how to segment it, and then I strongly failed on it and I left it for a bit and I focused on the biology part. I tried to segment them using some kind of like custom-made Python that was able in my head to get the curvature, but it was not, when you translated it to actual images, it was not so easy. So then, Amin from the Evers lab showed me how to use Cell Profiler and did the protrusion segmentation part, and I just found it so useful. It was so nice because in Cell Profiler you can just save the whole pipeline and do it step by step and export it automatically and really tweaks the parameters and understand how each block works. So it was honestly very nice. Now I'm a fan of Cell Profiler. This is I'm not getting paid or anything, but
it's very easy to use. We do have one question from the audience by Francesca Bottanelli. She says, "Great talk. Thanks for sharing. Have you tried to remove CD45 and assess if the interaction from the protrusion area and the whole cell membrane is affected?"
Sorry.
Francesca Bottanelli is asking whether have you tried to remove CD45 and assess if the interaction from the protrusion area and the whole cell membrane is affected?
No, no, I have not tried to do that. I mean, I'm trying to think if someone else has done it. No, but me myself, I have not tried it. But I mean, I know that the effect of like removing CD45 is that, I mean, it's a bit complicated because it also has CD45 also activates LCK indirectly. So if you somehow neglect it, you don't get activation. This is what I know about CD45, but I have not thought about how it would affect the close contact, like the protrusion contact formation. My guess would be that if you remove such an important protein, probably the protrusions would be less important. However, I mean, they're important not only because of CD45, but also because you have a lot of other proteins that are way bigger than the CAR or the TCR. So you will also have to like pass through them. But yeah, I have not tried it. That's my answer.
I think we are on time for our next speaker. Is it Helen? Or do we have? Okay, great.
I'm going
Thank
to
you.
stop it.
Thank you so much, Carmen, for your presentation. Amazing work.
Thank you.
We can answer the questions at the end as well. So yeah, we can come back.
Okay. Can you guys hear me?
Yes.
Okay. So, so I will continue where Felipe left about the preLights. So as a prelighters, we cover all the bioarchive categories, as you can see, which is listed here. So we write preLights post about all the different domains, animal behavior, cell biology, cancer biology, and so on and so forth. So it gives an opportunity to researchers from all the domains to be a part of the preLights and read about their research and write about it. So we do have an open call to join our community in preLight. And so there are many reasons to join preLights. And as I have been associated with preLights for almost 7 years now, I can tell you the most important reason is be a part of this friendly, diverse, and international community. And of course, you will gain a lot of experience in science writing and editing. And also you can build your own network and interact with the authors of interesting preprints like we are doing today. So anybody who's interested to join can send an email to prelights@biologist.com. And coming to today's second talk of this webinar, we have the first author of this interesting preprint targeting RhoA activity rejuvenates aged hematopoietic stem cells. And we have the first author, Eva, Dr. Eva, who will discuss her results shortly. So I will not take away any of the important conclusion from the study. I will let Eva talk about it. I will just highlight what I really like about this preprint, apart from the imaging, the interesting RhoA GTP microscopy images. So I really like how it connects the nuclear mechanosignaling with the image, with the aging of stem cells. And particularly, I like this linking of the KLF4 activation to actin polymerization. So it provides an explanation for how the cytoskeletal changes can reshape nuclear function and chromatin state. And I have a personal bias also for this article, because I also in my previous lab, I was working with RhoA GTP and migration in the context of caveolae. So I really like the work, and I will now let Eva present the work. Okay, I will stop sharing. Yes.
Hi, hello. Should I
Yes.
start sharing? Okay. I would like to thank also to the company of biologists for this opportunity, highlighting our preprinting in bioarchive and FocalPlane to make this webinar possible. So I will try to summarize what we have been working. We have been working in this period for this preprint, and the first is just to tell you that aging is we work on aging in HSCs, and we assess aging as the loss of regenerative capacity of HSCs. We know that in some other stem cells, alterations of biomechanical pathways lead to loss of stem cell function. And we were wondering if in the case of HSCs, that would be the way. We're focusing in RhoA in the hematopoietic system because there is little known about it. And the knockout we know is lethal. It provokes low number of HSCs and low differentiation in mice. And it affects cytokinesis in the during division, and with a final effect causing necrosis in the bone marrow. These are the key findings that I would like to walk with you during this talk, and they are that hematopoietic stem cells respond to nuclear envelope tension activating RhoA. We have seen that 8 hematopoietic stem cells suffer from physiological intrinsic nuclear envelope tension, showing high levels of active RhoA, and RhoA inhibition remodels chromatin and restores function in aged HSCs. So the first that we wonder is how an HSC in mouse looks like, and it's a small cell around 8 to 10 microns of diameter. Is a non-adherent cell, and it has a high nucleus cytoplasm ratio. This make us wonder how this cell that is not adhered to other cells or to the extracellular matrix is able to sense and to react to biomechanical exerts. We were inspired by the work of 2 labs that they published in 2020 in Science about how the nucleus can be a mechanosensor. This work was made by Valeria Venturini and Alexis Lomakin, and they did it in very different ways, but it's inspired us a lot. And we were wondering if this mechanosensory nuclear stretching pathway that they describe is also functional in hematopoietic stem cells. For doing so, we isolated HSCs from mouse, and we put them under a confinement device that allows us to have the cells under 3, 5, or 8 micron confinement. Remember that these cells are very small. We fix the cells under the device, and we analyze the physical properties of the morphometric features of the cells, also on the activation of the RhoA GTP under confinement. First of all, the diameter of these cells is increasing the more you confine the cells. The DAPI is showing much less intense the more we confine the cells. Also, if you look at the cells confined at 5 micron, you can see blebs that are consistent with the disruption of nuclear envelope that are even more strong in the 3 micron. And about the RhoA GTP, that is the surrogate that tells us that there is a mechanical stress in the cells, we see that the more we confine the cells, the more activated RhoA we have, seen as RhoA GTP in red here. At 3 microns, however, we don't see such an increase. In fact, we see a sharp decrease because we think this confinement is incompatible with life. To know if this activation of RhoA was actually a response for the nuclear stretching pathway, we checked how this pathway works, and it is that you have nuclear membrane stretching, you have the activation of phospholipase A2, which produces arachidonic acid, which in the end activates RhoA. So we stained in these settings for phospholipase A2 activated, and we saw that under 5 micron of confinement, we were able to see a huge increase of active phospholipase A2. This confirms that mechanical confinement activates RhoA, and the nuclear stretching pathway is actually functional in HSCs. But what about aging? We are interested in the mechanism of aging and using hematopoietic stem cell as a model. And since mechanosensitive pathways alter the system in a way that make some cells to age, we were wondering if it was the same for hematopoietic stem cells. And for that, we isolated HSCs from young and aged mice. Age is more than 80 weeks, as you can see here, almost 2 years. And we analyzed and measured the amount of RhoA GTP, the RhoA activated in aged cells. And we saw that physiologically, just cells on a cover slip were having an 5 times higher amount of RhoA than young cells. To know and to make sure that activation was specific, we also used a RhoA inhibitor, RhoSyn, to check for the not activation under the treatment with this, and of course we saw that this happened. Sorry, it doesn't, the statistical analysis doesn't show, but believe me, these and these are significant. We also measure the diameter as a physical property of the cells, and we observed that the diameter of aged hematopoietic stem cells was physiologically bigger than the young, and intriguingly, the inhibition of RhoA was decreasing the diameter of the cell. So we can conclude that RhoA is physiologically activated in aged HSCs. But how is this nuclear stretching pathway being activated in cells in the bone marrow that are non-adherent and they don't have any interaction with the extracellular matrix as other cells have? So we thought that maybe extrinsically, by changing in the bone marrow stiffness, can affect to this pathway and activate RhoA, but it could be also that it would be intrinsical. So in changes in the nuclear envelope tension itself that could. So for that, we realized we performed indentation experiments on bone marrow of young and aged mice. And we realized and we discovered that the aged endosome and inner marrow is much less stiffer than the young situation. So we can say that it's not an extrinsic effect of the bone marrow stiffness. It is something within the HSCs while aging that is increasing the nuclear tension changes. Again, just for you to remember, to make sure that the nuclear stretching pathway is on, we need to know if the activator phospholipase A2 is active and shifted to the nuclear envelope. We measured that, and we used an inhibitor for this protein also to make our observation specific, and we saw that there is an increase of the activated form of phospholipase A2 at the membrane in 8 cells that we are able to decrease when we use the inhibitor. And parallelly, when we use the same inhibitor for the phospholipase A2, we decrease the RhoA GTP. So we can say that RhoA activation is dependent on the nuclear envelope phospholipase A2 levels. And as a final conclusion for this part, aged HSCs are under physiological nuclear mechanical stress since their nuclear stretching pathway is active. But can we actually see these changes in the nucleus, in the nuclear envelope of the nucleus? And the answer is yes. For that, we use lamin B staining on young, aged, and aged RhoA inhibitor treated, because young cells have grooves and have foldings that we can see very well with lamin B, and lamin B levels doesn't change upon aging. We were able to measure the nuclear envelope circularity and the nuclear envelope excess of folding, meaning that, and we observed that the aged cells have much more circular shape. It's like a balloon that is being inflated, more than in the young and in the RhoA inhibitor treated. And for the excess of folding, aged cells have many less. which means that it's like in age cells, these foldings are resolved, but when we treat the cells with the inhibitor, they come back as a young situation. But how this is how this is happening? Something in the nucleus, the chromatin, is happening for this to happen. So we have used computational statistics and deep learning for DAPI images, only HSCs stained with DAPI. This part was made by Pablo Yáñez, a predoctoral researcher at IS Global under the supervision of Carolina Florian and Paula Petrone, for studying how the chromatin is changing upon aging, and if we are able to modify and remodel with our inhibitor, the RhoA inhibitor, this chromatin organization. He used features, and he extracted features from 3D and 2D images from our HSCs. The features resemble somehow the changes that we were seeing when we were stretching our cells at the level of 8 micron. They were going in the same direction, like the volume or the surface area, for example, in the 3D, or in the 2D, the largest slide perimeter. So we concluded that chromatin remodeling upon aging is comparable to mild mechanical stretching. Not only that, we were extracting a lot of features from DAPI HSC's stained cells, and we were able to compile 20 features that were able to give us different but yet similar patterns that we can compare the same way that we compare RNA, single-cell RNA-seq. So Pablo was able to construct a UMAP in which we could see how, depending on how the cell was, either an age, young, or treated with RhoA, it could fit in a different place in the UMAP. In the end, we could conclude that chromatin remodeling is associated with RhoA inhibitor treatment on aged HSCs. But how these big changes in the chromatin that we can see in DAPI images are made? For this, we did ATAC-seq and look for the molecular changes to look at the accessibility of the chromatin within HSCs. And we confirmed, as it was already known, that aged cells had a global, more open chromatin than the young. Intriguingly, aged cells treated with the RhoA inhibitor were coming back to the situation of the young. It was less accessible. But if we focus a little bit more, it was very interesting that repetitive elements within the chromatin were, as line and LTRs, were the ones that were more open in aged and more closed when aged were treated with the RhoA inhibitor. Here you can see the levels of accessibility that we were losing in compared with the aged. To confirm that, we did also ATAC-seq in confined cells, 5 microns, and the specific places where we saw differences were again in repetitive elements as line and LTRs. So when we confine young cells, the sequences that we found more open were corresponding to these repetitive elements. So which parts of the chromatin are we changing? You know that the chromatin can be divided in heterochromatin and euchromatin. And one of the marks for heterochromatin, that is the more compacted chromatin within the nucleus, is H3K9 methylation 2. Then we used this mark to stain young aged and cells aged treated with RhoA inhibitor, and we discovered that aged cells have a very low staining of H3K9 methylation 2 compared with the young, and when we treat the 8 cells with the RhoA inhibitor, we restore the levels of the heterochromatin mark, which means that RhoA inhibitor restores young level of H3K9 methylation to unaged HSCs. To know which methyltransferase, and you know that are many, and it could be a lot, but we checked in G9A and we used the inhibitor UNC of G9A, and we saw that it was the activity of G9A was required for the restoration of H3K9 methylation 2 when H cells are treated with a RhoA inhibitor. To genetically prove that the heterochromatin loss is caused for nuclear swelling that caused the stretching, we engineered a retrovirus containing form of H3.1 gene, which is not able to be methylated. So the normal one is K9 that has a lysine, and the mutant has a arginine instead, and it cannot be methylated, so it cannot have heterochromatin. We infected progenitor cells, hematopoietic stem cells and progenitor cells, and here you can see how in the R9, in the mutant, there is no H3K9 methylation mark. When we check in long-term HSCs about the activation of RhoA, that is a surrogate for the activation of the nuclear stretching pathway, we observed a big increase in the activation of RhoA. And not only that, we also saw how the nuclear volume was increased when the heterochromatin cannot be formed in the R9 overexpression. And about the function, once you transplant stem cells that are overexpressing R9, we observe low engraftment both in the bone marrow and the peripheral blood, and also unbalanced differentiation, which in the end means that these cells that lack heterochromatin mark H3K9 have a low regenerative capacity. And also, we did a study how these changes in organization would be affecting into the gene expression. And for that, we did first bulk RNA-seq on the cells to check how the RhoA treatment was affecting changing the gene expression in H cells. And here you can see that we were able to downregulate genes that are normally upregulated in aging, like myeloid cell activation, response to interferon beta, or acute inflammatory response. And if we check in gene expression signatures, they were down after the treatment with the RhoA inhibitor for aging, acute inflammatory response, and interferon. So we could confirm that the RhoA inhibition in HSCs downregulates aging and inflammation genes. If we focus on specific genes, we were intrigued because we saw that the KLF family genes are upregulated after the treatment with RhoA inhibitor in HSCs. To confirm and to explore a little bit more about the gene expression after the treatment with RhoA inhibitor in HSCs, we did single-cell RNA-seq out of LSK that contain the HSCs cells and also some progenitors. So we did the single-cell RNA-seq, and the results were analyzed, and we were clustering the different population, and we observed that the only population that had upregulated KLF4 was HSCs in age treated with a RhoA inhibitor. We also were confirming what we saw before, the inflammation genes and the aging genes were also down when we treated the cells. But very interestingly, and using the tool Scenic, we could see that only the aged HSCs treated with a RhoA inhibitor were activating KLF4, and all the transcription factors down in the pathway of KLF4. And since KLF4 is one of the genes that is used for reprogramming of somatic cells, we were wondering if there would be some kind of reprogramming. So we checked for hemogenic precursors, so the genes that are expressed right before the cells are hematopoietic stem cells. And we were right, the hemogenic precursors are upregulated in H cells that are treated with the RhoA inhibitor. And what about function? Are all these chromatin organization, gene expression, changes that we are seeing being translated in a recovery of the function of HSCs? So for that, we did a transplantation assay with H cells and H treated with the RhoA inhibitor with a young as a control. And we observed that the peripheral blood engraftment at 18 weeks was restored in the H cells treated with the RhoA inhibitor. And also the B cells were restored together with a decrease in the myeloid cells. So we are actually seeing the opposite of what we see when we are when the cells are aging. We are recuperating and restoring the regenerative capacity in aged HSCs. So to summarize, I would like to have this cartoon. I didn't say about I didn't talk about the actin itself, but we saw that after the treatment with the RhoA inhibitor, the actin expression in aged cells was restored compared with the age. So yes, there is this link in between how we are able to reprogram to a more functional HSC that involves several restorations of actin, and TAS, for example, was also restored, and so on. But the final picture is that the chromatin fitness is improved, as we see with H3K9 methylation, heterochromatin mark increase. We close the line and the LTRs, and the inflammatory signature is down. About the KLF4, we see an increase that we don't see when the cells are young, but is something that is happening only by chemically targeting RhoA. So we are able to maybe chemically reprogramming HSCs. And last but not least, the hematopoietic function is improved and is recovered, as we have high lymphoid and myeloid ratio, and we have an increased engraftment. So this and more you can see at our preprint in bioRxiv, and I would like to thank everybody in the lab, in special Carolina, for giving me the opportunity to perform this project. It has been very challenging, and I have learned a lot. I would like also to thank the other first co-authors, that are Pablo Yáñez and Bárbara Werther. Pablo has as a mentor also Paula Petrone, that we have discussed a lot of things. We have learned a lot, and I would like also to thank everybody you see here involved that is involved in this publication. So we have to, I want to thank you also, and I'm open to questions.
A very interesting talk, Eva. So do we have more questions from the... Okay, so we have a question from the attendees that Annapurna Sharma is asking, is saying, insightful. Do you plan to look at the chromatin interaction using techniques like HIC to know more about the changes at chromatin levels, TADs and loops?
Yeah, the HIC, I think he means HIC.
Yeah,
yes. It's in fact, I was trying to do HIC, but I have to underline that all these genomic analysis we have been doing, it has been with a very low amount of cells, in between 1,000 or 2,000 in the case of bulk RNA-seq, and in the case for ATAC, it has been 2,000. So we are working on it. We are almost there with the high C, but knowing how is the changes in the chromatin, it will help us a lot. Of course, we also can merge. We can merge single cell ATAC-seq and RNA-seq, for example, and it will give us a lot of information about the organization itself. But we are very interested, more than the which is close to what, is that we are able to chemically, with an inhibitor, being able to modify the chromatin in a way that is restoring the function. So yes, of course, it would give us a lot of information, but we are intrigued in all the steps that we are still missing, like the mechanistic connection that we don't know yet, and to be sharper in how we can manipulate these changes in the chromatin. Yes, of course, we can do it with a RhoA inhibitor, but maybe if we dig a little bit more in the pathway, we are able to do it also in different ways. Because RhoA inhibitor is very difficult to, for example, to imagine inject in the mouse, because the life of the chemical compound is very low. So it doesn't reach. So it would be nice to know what is underneath and what are the steps that we are missing.
Okay.
Thank you.
I have some questions, but I will probably ask you when I write about the post, so it will be much more easier. Do we have more question, or should we move to the discussion part?
I think let's move to the discussion. And if people have questions for either Carmen or Eva, keep typing them in and we'll pass them on and we'll pass them to them, and then we'll publish it with the recording, any additional questions you might have.
Okay.
So I guess, Felipe and Vibha, did you have some general points you wanted to raise?
Yes. So is a very generic or general questions about preprints. Like so, I wanted to ask, particularly in, say, in your institute or in your lab, do you guys always like prefer to post it on bioRxiv, or do you also read and discuss preprints in your like literature club and so on? Or how frequently are like preprints being discussed as a part of your journal club or something?
I have to say that in our case, preprints are not usually taken for our journal clubs. We've been surprised, I have also to say, about the reaction that we have had with the preprint, because we have had a lot of positive reactions, and it was the very first time that Carolina's lab has posted a preprint, and we are very happy about how it came, and it will probably make us consider to take the preprints as an option for journal clubs, of course. And I was also very happy when I saw, before you selected us for this talk, that you were already making a filtering of some highlights. So that was very useful, because I saw a couple of them that were already interesting, and I was like, oh, these people is doing this, so I'm very happy about the experience.
I think Dr. Bottanelli has some shared opinions on this.
No, I mean, I love preprints. It's great, and it, it gets the science moving faster, the sharing of the science, and you also have some kind of sense of achievement after you publish a preprint, right? Rather than put it in the pit of peer review, which It's a long tunnel that never ends. It's kind of like, right? Carmen, we always celebrate submission of preprints.
Yeah, I didn't talk about the preprint decision.
Oh yeah.
It's
Yeah.
also something to consider. But since that moment, I'm super happy.
Yeah, and also, I mean, it's great to discuss preprints in journal clubs because you know they haven't gone through peer review and but I must say that okay, now we're getting philosophical, but this but you know, I feel like that peer review is a little bit of like, you know, a lottery. It depends who will get your paper and who will tell you to do this versus other people that have strong opinion on that and then will tell you to do that. And yeah, and in a lot of cases, obviously, it makes the paper better. It makes you, you know, strengthen some aspects that maybe you hadn't thought about. So maybe it's nice to look at those aspects before somebody else, 2 out of 3 people have thought about it.
I also wanted to ask, like, question to everybody that has anybody ever contacted the author after reading a preprint and maybe like just directly emailing the corresponding author to give some input or some kind of questions or something, which may help the authors to like either improve or add some more experiments and things like this?
Not in our case.
Yeah, I have personally done that before, and it led actually to a collaboration with an international group and my previous lab. So it's a great experience, and
Yeah.
people should take advantage of how fast-paced
Yeah.
the pre-printing scene is. Is very useful if you can take a little bit of time to contact authors, just to let them know, even
Yeah.
if you liked their work, that is also very
Yeah.
good.
Yeah, in my case also, I have, I mean, I definitely benefited a lot by contacting the preprint authors and getting the reagents before the paper was published. And actually, I used it for my study. So it was really, really good experience. So, yeah.
Nice.
I mean, people have done it with us. They've sent, hey, I've read your paper. It's great. Have you thought about this? Have you thought about that?
Yeah.
And I've done it with other people as well. You know, hey, I've just seen your preprinters online. This is great. But
Yeah.
yeah, it's yeah, it's cool.
So I'm just asking the audience the same question, but carry on. I don't know, Felipe, do you have anything you want to
I
ask?
think Vibha covered almost the same questions I had, but in overall, do you see any case in the future where you really more advocate to promote pre-printing culture at your labs or any public appearances or conferences? How do you plan to keep moving this forward so that not only peer-reviewed articles are getting the highlight on everything?
Since it was my first experience, I cannot I cannot say anything, but it would be I mean, something to repeat.
I don't know. I have a feeling that some people are still, you know, they still don't post preprints because they think they'll get scooped. Instead, that in my opinion is kind of like a scoop protection, right? Because once your
Indeed.
preprint is online,
Yeah.
it's out. You know, it's not peer reviewed, but it's kind of out there. So you've told the world that you've worked on that.
Your work. Yeah.
That's
Yeah,
an
I
excellent
personally
point.
know people who are who have the same issue that, you know, once we put it on preprint and by the time it's published, we might get scooped, which I also didn't understand the whole point.
So you're actually time stamping your idea.
Yeah.
Like,
So I'm glad that there is actually communities that actually promote preprints and yeah, I think it's very cool. what you guys
And
are doing.
company of biologists and preLights is actively recruiting new prelighters. Yeah, so I guess Alan can do a better job at advertising that. But in general, you can join at any moment. The team is amazing. They're very welcoming, and it's overall a great team to be part of. So
On this
I'm
wonderful,
grateful.
sorry, I think in this wonderful note, I would like to say just one word and spotlight the fact that behind the preLights, there are wonderful community managers. I'm not talking about myself because I'm here temporarily, but I know people like Helen and Rainier, who is coming back after leave. And so these people are really taking care of the community and sort of helping to sustain a nice environment and to provide a nice feedback on the pre-lights and just like to create this nice interactive chemistry between the people who join as pre-lighters. And so, yeah, saying that, I also wanted to thank Felipe and Vibha for stepping in because initially it was sort of a trial project for us, kind of an idea to try things out. And then we reached out to you guys, and you were so happy to step in that it immediately gave us a lot of inspiration. But then we also want to thank the speakers, so Carmen and Eva, and also Francesca, because yeah, as you guys discussed, so preprints is yet not the most widespread currency in the scientific community, and therefore it's really nice to find people who are willing to talk about it, who are willing to sort of share their experience around it. So yeah, thanks a lot to all of you. Thanks to people who joined in as attendees, as listeners. And one more time, send your questions, and we'll share with all the speakers and hosts. If any of you have any feedback on this format, we'll be happy to hear your thoughts or read your thoughts. You can send us an email on prelights@biologist.com. You can see it now in the webinar chat. And yeah, do we have anything else? No, I don't think so. So just wanted to thank you one more time. A wonderful day, morning or evening or afternoon, whatever that is for you. And yeah, hope to see you around, particularly on the pre-lights and the focal plane.